Well, hello, micro friends. So we're moving on from bacterial diseases, getting into a really cool topic of particular interest right now, and that is the biology of viruses. Now, viruses are obviously, or maybe not obviously, they ain't bacteria. Viruses are pretty weird little bits of information, and their life cycles are quite varied, and they are important disease-causing agents. Treatment of viruses is limited because we don't have antibiotics. We have antiviral agents, but they don't work all that well, and they're not all that general. And we're living through a viral pandemic as we speak. So let's get into it. We're going to be getting into today and this week is characteristics of viruses and sort of viral life cycles and stuff. And then we're going to start getting into some of the diseases of viruses and kind of work it the way we did with bacteria, different routes of entry and different kind of viral diseases. So just to begin with, we have some basic vocabulary. Viruses are really just little bits of information. I mean, the term computer virus is really quite apt in that, in a computer virus, you're infecting the computer with a little bit of code, a little sequence there. And the same thing is happening with a cell, is a virus is putting in a little piece of nucleic acid sequence, the pieces that are required to replicate itself. So we have a couple of things that are always present and kind of required for viral operation. Firstly, we've got, excuse me, the, hold on, pointer, there you go. We have some kind of nucleic acid. The nucleic acid might be DNA, or it can be DNA, or it can be RNA. And honestly, when we start looking at viruses, we can have double-strand DNA, which is what, of course, is in bacteria cells and in our cells. We can have single-strand DNA. We can have an RNA genome. We can have double-strand RNA or single-strand RNA. And additionally, we'll see we can have what's called a plus-strand RNA, which is an RNA that is ready to be translated. It's like what would be generated off of a gene. And we can have minus-strand RNA, which actually has to be transcribed in a different way, and all kinds of interesting things that viruses do that really don't ever happen unless you have a viral infection. And so some pretty interesting kinds of phenomenon and biology are involved in viral life cycles and the way viruses work. And they are a organism worthy of intense study. Now, whether they're alive or not, I would say it's debatable, but not really. They're not really living. They are obligate intracellular parasites. They don't have the ability to actually carry out the production of proteins from their own genomes. They can't transcribe the genome. They can't take the RNA, or even if they're an RNA virus, and translate it. They can't produce any of the stuff required to assemble themselves. They have to get into a cell, and they're going to hijack the cell's machinery to replicate themselves. And so, in order to actually bind to a cell and inject its nucleic acid or get the whole virus into the cell, we have to have some kind of receptor interaction, and that the virus is going to have a receptor on it that allows it to identify and bind the host cell. Just in the case of the novel coronavirus, the COVID-19, it binds to something called the angiotensin converting enzyme receptor. This is a hormone that's involved in blood pressure regulation, and it's also involved in lung interactions and things, the way the lungs are operating. And so the receptor that the virus binds to, or the protein the virus binds to, is found a lot in the lungs. And that's what's going to actually determine what cells are going to get infected by a virus. Some viruses are going to infect very specific cells in the nasal mucosa, in the lungs. Some viruses infect specific cells in the immune system, like the virus that causes AIDS. Some cells or some viruses will infect neurons. So it has to do with how the virus actually interacts with the proteins on a specific cell surface. So they pretty much are inert outside of a cell. That is, they can't replicate on their own. They are much smaller than cells, and so you can filter out cell debris and then still have viruses coming through. They are non-living. They are, in fact, quite diverse. Probably in terms of evolution, viral evolution, different viruses have come about in multiple times during evolutionary times since really the very first cell. As we're going to see, bacteria have viruses that infect them. So here's just some examples using electron microscopy. These viruses are quite small. They would be completely invisible under a light microscope. We've got things like the tobacco mosaic virus, which is actually a plant virus. It is a helical rod shape. The polio virus has these cool icosahedrons, or in a cross section, these hexagons, and it's referred to as being a naked virus because it doesn't have a membrane around it. Herpes virus is kind of a blob. It's actually enveloped in that it has a membrane around it. And part of this is going to be as the virus is going to spread from the cell. Some viruses will just pop a cell open and dump the viral particles out. Other viruses actually are going to be caught up in a vesicle and be secreted by an infected cell as membrane-bound viral particles. Let's see. Here is a pretty large virus called the Marburg virus, which actually is one of the viruses that causes Ebola, or it is like the Ebola. It causes hemorrhagic fever and bleeding and stuff. It's got this really cool squiggly structure to it. Here's a smallpox virus. It's actually what's referred to as a complex structure. We'll see that in just a moment. It has a variety of different proteins on its cell surface. So viruses come in a range of sizes. We have the Ebola virus, which is a really pretty big guy, pretty long anyway. The variola virus, which is causing things like smallpox and stuff, pretty big. Here's herpes virus, rabies, measles virus, little tight guy. HIV is a small one. Here's one we've been talking about, or you've been hearing about, the SARS virus. This is a coronavirus, very much like COVID-19. Adenovirus causes some colds and things. Rotaviruses cause viral diarrhea. Influenza virus. Then we got some real small guys. And one thing is the genome size changes as well. Some of them have pretty large genomes. Some have very small genomes. Things like parvovirus, which is actually a virus that's important in vet med because it infects dogs. Hep A, Hep B, Hep C. A virus actually we've been working on in the lab, the Zika virus. And papillomavirus, human papillomavirus, HPV, is involved in various sorts of cervical cancers and things. So we'll get into this as we continue. But lots of different sizes. Now, you can filter out bacteria, but viruses are, in fact, able to pass through filters. So here's an example of viruses that are sort of blobbed together. This is a norovirus that was found in a cruise ship that was responsible for massive diarrhea and food poisoning. One reason, if you're going to go on a cruise, you probably should bring your own food. But viral induced food poisoning is something that absolutely happens. And when you pass a liquid, contaminated liquid, the bacteria will get caught on the filter. But the viruses are too small. They will pass through. And so that's one of the ways you can decide if you're working with a pathogen that is a virus or a bacteria, is if you can filter it out, it's a bacterium. If you can't filter it out, it is quite likely a virus. Now, you can also see viruses by the way they interact with cells. Now, on the left here, this is a bacteria plate. And these spots are known as plaques. And a plaque forms when a single cell is infected with a bacteria virus. Something's called a bacteriophage. And as that bacterium that's been infected replicates and just divides sort of in a bilateral way to create a colony, a circle, when that virus then kind of decides it's time to go ahead and pack up and blow the cell apart, you'll have this circle of, or this dot of cells that will all lyse more or less at the same time, and you end up with this halo, this clear spot known as a plaque. Now, this is another kind of viral plaque assay using the vaccinia virus, and this is using not bacteria, but some kind of animal culture cell, and you still see the same kinds of plaques where the virus is that has infected the cells blows the cells up. Viruses can be mean and sneaky little dudes. You never want to turn your back on a virus. So we can classify viruses in a couple of different ways. We can classify them in this style, in which we've got double-stranded DNA viruses, single-stranded DNA viruses, double-stranded RNA viruses, and then single-stranded RNA viruses, either the plus strand, just in terms of the biology, plus strand indicates that this is the RNA that is capable of being immediately translated to produce proteins. The minus strand is actually the complement to the plus strand. And so the virus, in order to carry out its life cycle, has to have an RNA polymerase that will code a new RNA that will be the plus strand. And then you have single-stranded RNAs that use something called reverse transcriptase. These are RNAs that are actually converting themselves into a DNA molecule. Reverse transcription is the term we use for when we take an RNA and, using an enzyme called reverse transcriptase, turn it into a DNA. Now that's actually backwards from our dogma of DNA is transcribed to RNA and RNA is translated to protein. In these cases, they're taking RNA and transcribing it back into DNA, which is pretty bizarre, but that's what viruses do. Then there are some double-stranded DNA viruses that also use reverse transcriptase in various complicated ways of making their the organism. They produce RNA and they convert that back into DNA. Hep B is one of those. So viruses, in terms of molecular biology, they pretty much represent every possible scenario you could in any way invent some kind of information exchange. There's going to be a virus that does it. So another way of classifying viruses is through their portal of entry. And from a pathological or from a pathology perspective, this may be actually more important. What the virus is, its genetics are, may not be nearly as important as the kinds of things it's causing and the ways it's being transmitted. So we have food, waterborne viruses. These are getting into the mouth, through the nose, into the GI tract. Very often they're going to be in contaminated water contaminated by fecal material. Please don't. Airborne, these are things that are coming into the mouth and nose, and now they're getting into the respiratory tract. Now, again, the virus is going to infect cells that have got the appropriate proteins on their surfaces for the receptor to interact with. So, airborne viruses, like COVID, these are most commonly brought into your body by inhaling air contaminated by a sneeze or a cough. One of the reasons why wearing masks is so useful is you're minimizing the amount of virus that you're dumping out. Sexually transmitted diseases, these are generally pretty delicate viruses, and they actually are not very good at living on cell surfaces and things, or living outside of their environment. Other kinds of classification or portal of entry is what's called cross-cutaneous, in which we've got receptors that are on the internal cells, but the viruses are being found in blood and contact, and they say have to get into some kind of wound, or somehow get into the skin, or get into the bloodstream, like vector transmitted type viruses and things. So this is another way of classifying different bacteria, or different viruses. All right, so some basic components. You have what's called the core, which has the nucleic acid. You have the capsid, which is all protein. And then there's going to be things like spikes and stuff that are receptors. And then there is the envelope, and this is not always present, but often. The shape of the virus can be useful in terms of identifying what kind of virus it is. And so we have these things like the terms we call icosahedral or helical, binary or complex. And we'll look at them here individually. So this is an icosahedral virus, the adenovirus. This is what causes some colds. Rhinoviruses cause other colds. There's even a few colds that are caused by coronaviruses. So the adenovirus has the DNA inside this protein structure, this protein capsid structure. And then there are these spikes that are going to interact with proteins on the cell surface of the host. Now, this is referred to as a naked virus. It ain't got no clothes. It's not membrane bound. That's what the term means. Now, the opposite of being naked is to be enveloped. So the herpes simplex virus and chickenpox and a whole range of them, they actually have a membrane surrounding them. And this membrane has got a series of glycoproteins on them that are part of the envelope. There's an inner layer of protein inside, and then the capsid is the protein surrounding the nucleic acid inside this membrane. And so I sometimes think, we can kind of imagine viruses as being a little bit like a corn dog, and a variety of different kinds of corn dogs, where you've got the hot dog is the nucleic acid, and then you got the cornbread surrounding it. But it depends on the different virus types. Some of them have membranes, some are just coated with proteins, different kinds of viruses. Filamentous viruses, these are primarily not pathogenic type guys, but we've got the tobacco mosaic virus, which has got a whole set of proteins surrounding the coiled RNA. Now, an enveloped structure like the influenza virus has got the lipid bilayer surrounding it, and then there are a range of proteins on the cell surface, and then deep inside we have the RNA molecule complexed up with some proteins to kind of give it some structure and protection. So viral shapes continued. We've got the filamentous type. We have the icosahedral type. We have the enveloped type. Mainly they have a plasma membrane around them, a double bilayer. They didn't biosynthesize it, they just are kind of coated in it. And then this term we refer to as the complex type, where they've got multiple protein structures. This guy looks a little bit like some kind of spaceship lander, doesn't it? In that we have a head, which is in fact an icosahedral structure. The DNA is inside, and this is an example of a bacteriophage, specifically the bacteriophage T4. And it's got this lower trunk and then these kind of little feelers. These are the spikes that are actually going to interact with specific proteins on the bacteria's surface and allow it to bind, and then the DNA is injected into the cell. This whole structure, in this case, is going to stay on the cell surface, so it doesn't get incorporated into the cell. So a lot of what we know about viruses and sort of the definitions and ways of thinking about viruses we've gotten from bacteriophage. So these are the viruses that can infect bacteria, and there are a range, a wide variety of them. These were discovered back in the 40s or so, and you can identify them from sewage. You can identify them in a lot of different spots. There's all kinds of bacteriophage. And you have a bacterium and a specific virus that infects that bacteria. And the virus can be very, very specific to individual strains or individual species of bacteria. So E. coli has got a set of viruses, and Klebsiella has another set of viruses, and they don't even cross-react. So a lot of the way we talk about viral life cycles and the terms come from studying bacteriophage. So, the first step is you grow up some bacteria, and you then take a solution that's got virus in it and shoot that into the bacterial culture. You can spread it on a plate, or you can put it into a liquid culture, and you let it sit for a few moments, 10 minutes or so. That allows the virus to actually bind to the cell surface of the bacterium. And next is the viral genome is being squirted into the cytoplasm of the bacterium. This is the penetration step. Once the nucleic acid gets into the cell, it is then going to actually serve as a template for the production of RNA and then the translation of that RNA into proteins, and you begin to synthesize all the little parts of a virus. So you'll synthesize the proteins involved in the head, the proteins involved in the tail, and at the same time, the nucleic acid is also replicating. And once things start to get enough parts and things together, it'll autocatalytically begin to assemble themselves. And so it's a little bit like having a bunch of clothes floating around in the swimming pool, and then you throw in a whole bunch of open suitcases, and the clothes start to automatically interact with the suitcases. Boom, the suitcases close up, and you've got these infectious viral particles again. And we can think of it as sort of magic and autocatalytic ideas. What it means is that the proteins actually interact with each other in a way that will attract the DNA, and the DNA gets coiled up inside. And so you begin making a lot of bacteria viruses. So one cell is going to be able to synthesize thousands of individual viral particles. And eventually, as the cell starts to slow up its metabolism, or cell division begins to slow up, the viruses decide it's time to go ahead and bug out. And they will pop the virus and release all these viral particles into the surrounding media to infect more bacteria. And let's say, not that we want to anthropomorphize, but viruses are fairly evil. They have one job, and that is to create themselves and to mess you up. Never trust a virus. Sorry. Viruses are going to do what they do, and what they do is pretty much tear a cell up. So you may have a virus, a pet virus, that you think you know, and you pet it and you feed it and you are really talking nicely to it. Don't turn your back on that virus, because it's going to hurt you. All right, so we have lysis. Now, another thing that the bacteria can do is actually integrate itself into the host genome. And this is just another diagram of what's known as the lytic life cycle. So with bacteria, or with viruses in general, we have these two different ways of thinking about their life cycle. We have the lytic phase, in which the bacteria or the host cell has got the virus, is replicating it and allowing it to produce more progeny and begins to assemble them. And then the lytic part of this means that the cell is going to begin dumping the virus out into the environment, either through a membrane, through sort of membrane vesicles being pinched off, or in the case of bacteria, being ruptured, killing your host. And that's the lytic cycle. So we've got attachment, entry of the nucleic acid. The phage DNA is going to begin synthesizing. The phage proteins begin synthesizing. We have assembly, and then release. And then this whole evil cycle repeats itself. Now, with hepatitis, now this is in a eukaryotic cell, we have the nucleus right here, and the virus attaches. It's brought into the cell through this notion or this mechanism called endocytosis. That is, the membrane sort of engulfs the virus at the cell surface and pulls it in. Didn't mean to do that, because now you have let the evil one into your house. So, as we're going to see here, one of the viral treatments is to inhibit the virus from landing on your cell surface, and then you don't pull it in. So basically keep the virus from knocking on the door. There are other mechanisms that would be possible, like you could inhibit the, quote, opening of the door, but that screws up a lot of other things that you need to do, because we often are bringing things in all the time. Viruses are just kind of tricky that way. They ring the doorbell. Hi, I'm just a molecule on your cell surface. Bring me in. You bring them in, and then it's too late. Great. So, in this case, the virus gets in, and it begins to produce RNA. The RNA is translated, and the RNA products, the proteins, begin to package themselves up into infectious viral particles, and we then have release as the virus is being now passed back out into the cell. So, another aspect of the viral life cycle is really insidious, and this is what's known as lysogeny. This is a condition in which the virus and the viral genome is incorporated into the cytoplasm. It's penetrated in, and it gets brought in. And now the viral genome will integrate itself with the host's genome. And so the host goes, oh, a little piece of nucleic acid. Sure, land right here. And now I will just start to replicate you along with the rest of my chromosome. No problem. I don't mind. Come on in. And it's a little chunk of nucleic acid. Not going to bother anybody. And what you're doing now is every generation, your progeny have the virus in their genomes. And as I say, viruses are evil. Don't turn your back on them, because at some point the virus is going to say, hey, I'm happy to be incorporated into your genome, but I'm going to do what viruses do. I'm going to pull myself out of the genome, begin generating a lot of myself, generating all my protein structures, packaging myself up, blow your cell apart, and begin to go on to infect others. Oh, thanks a lot. After all these replications that I was so happily doing for you, how are you going to pay me back? By blowing my cell apart. Lysogeny. What lysogeny is doing is allowing for the host to replicate and carry on the virus without actually blowing itself apart. So we've got, starting over here on the right side, we have the phage attaching to the receptor on the bacteria cell wall. The DNA gets inserted. The DNA now is going to get inserted into the genome of the bacteria. The bacteria then replicates by binary fission, and this can continue on and on and on. And you're just making more and more of the progeny are carrying the virus. But then something's going to happen. Like in the case of the bacteria, you'll start to run out of food, or you start to get too crowded. Something will start to limiting the way, the speed at which you can replicate. At that point, the virus goes, hey, been nice, but I'm packing up, and it heads out. In animal cells, the lysogeny can also occur, and the trigger for this is often stress. So, lysogeny is a very creepy way of having a latent virus floating around. So, in humans, a really good example of a latent life cycle is found in the herpes virus. The herpes simplex virus, or HSV is known. The herpes simplex virus is actually has the ability to incorporate itself into the host's genome. And so you have the host genome, and then this HSV genome is packaged up and kind of associated with it. And the cells can divide and continue producing cells that are carrying the genome. And then at some point, through stress or other kinds of triggers, you'll have those cells now carrying the viral genome begin producing active virus. This is what generates what is known as an acute phase. And so herpes, like a mouth sore, it actually is being held in the neurons. And then during stress, or like you bite the inside of your mouth, or any number of things that can trigger a herpes cold sore. The cold sore manifests itself as the virus no longer being in the latent portion of the life cycle, but actually coming out. And so this is how we have reoccurring cold sores. So the virus is present in the genome of these neurons, and then because of different stressors, those cells that have the viral genome, they go ahead and express the virus comes out, and you have a little cold sore. So what you can do is treat the cold sore, but you really can never clear the virus from the host genome. You're stuck with it. So viruses are assembled. They don't divide. They are being manufactured. And this is a way of looking at number of infectious particles in that using bacteriophage. And so we've got a early phase infection in which not much is happening. And then you begin to have a lot of bacteriophage being generated inside the cell. The cells begin to accumulate the virus. And then at some point, some of the cells begin to pop open, and we now have extracellular viruses being produced. So once the virus takes hold of a bacteria culture, they tear it apart, man. It is going to be bad news for that bacteria culture. So, as I said, or we've talked about this, antibiotics don't work on viruses. And, this is the common sort of the issue when people come into the clinic and they've got a cold and they may want an antibiotic. Now, viruses are going to place a strain on the immune system. You become immune compromised because you're fighting off the virus. That can allow a secondary bacterial infection, sure. But the initial infection, if it's viral, antibiotics aren't going to do a thing for you. So antiviral drugs are always there in the process, and certainly COVID-19 has been a impetus for developing more antiviral drugs, but they're not real effective, and they can often be very specific to very specific viruses. So the goal is to inhibit viral infection or inhibit viral replication, but not screw up the cell. Now, one of the things with antibiotics when you're working with bacteria, because prokaryotes have a whole lot of things that are so different than eukaryotes, it's easier to target things like the production of the cell wall or the way the RNA polymerase works, the way the ribosomes work. And so we end up with antibiotics that way. Antivirals are a lot harder because the system itself is you. And so you want to inhibit protein production that's going to inhibit protein production in all your cells. What you can try and do is disrupt some of these viral specific things. So it's hard because the virus is using the machinery that we use. And so it's really easy to disrupt a cell or disrupt a virus by killing the cell, but that ain't very comfortable. So we have some successes for sure. HIV, the virus that causes AIDS, turns out there are inhibitors of the reverse transcriptase. The drug AZT does this. Protease inhibitors are also able to block the ability of the proteins that are produced to assemble, because the proteins are produced as sort of a large structure that has to be proteolytically chopped up into smaller pieces to be able to assemble. You block that protease inhibitor, and the normal cell functions don't require proteases like this, and that will block the ability of the virus to replicate. HSV, the herpes simplex. There are some polymerase inhibitors as well. Ganciclovir or acyclovir. This is a standard drug now, actually over-the-counter drug for herpes simplex. So when you get the feeling of a little cold sore, you can put a little drop of acyclovir on there, and you're blocking the ability of the virus to replicate itself, and so that can help fairly well. Tamiflu is used as a treatment in the flu, and it prevents the bacteria or the virus from uncoating itself, which is necessary for it to release the genome into the cytoplasm in order for it to get taken up or used to replicate more viral particles. It works kind of. What Tamiflu generally does is it'll shorten the flu, but it doesn't completely block it. Probably the best place to focus on viral or antivirals, this is vaccinations. Let the immune system do the work. Vaccinations work really well for viruses. And in fact, often that is the only antiviral treatment that you have. Some viruses, it's hard to make a vaccine to. HIV, there is no vaccine for HIV. But for a whole range of other viruses, we got really good vaccines. So in terms of natural defenses, we've got a variety of different kinds of systems. First off, some of the same things we talked about with bacteria is we have the epithelium, which is a really good barrier, your skin. The epidermis is dead cells, and dead cells can't replicate a virus, so that works. You can have cells that don't have the receptor. No receptor, no attachment, you can't get the nucleic acid in. There are always proteases that are floating around in the blood serum, and these can digest the protein coat and release the virus, just the naked RNA or DNA, which is going to get chewed up really fast. There are a range of intracellular nucleases, because there's going to be a period of time where the virus opens itself up into the cell and releases the DNA or the RNA, and it can get chewed up. That can work. Interferons are hormones. These are proteins that are released by infected cells that are kind of indicators of viral attack, and they can, in a sense, set up systems to inhibit neighboring cells from getting infected. Leukocytes, these are like white blood cells, and they are able to recognize cells that have been infected with a virus, and they can destroy them. Then we have the kinds of things that you're triggering with a vaccine. That's what's known as adaptive immunity, and hopefully we'll be talking a little bit more about that as we go on. This is the production of antibodies and lymphocytes. These are cells that are both antibody producing and involved in the immune system, and they're able to identify viruses and destroy them, and they can also destroy virally infected cells. So if you've got a good immune response, then the virus isn't going to be able to do anything to you. Now, one of the ways you get this is by having a viral infection and developing an immune response, and then you're more or less immune, hence the name. When the next time you see that virus, it comes in, you've got a whole range of proteins and cells that can attack that virus and pull it out of solution. As an example, one of the things that it's apparently somewhat successful with COVID, but it's actually very successful with hepatitis, is something called a gamma globulin shot for hepatitis biz. What this is you inject somebody with antibodies against that virus. And if you've been infected, and you know you've been infected recently, in a matter of hours, days, you can inject a selection of antibodies that'll then go through and scavenge up the virus and pull it out of solution and pull it out of the bloodstream, and you don't have an infection much anymore. So that works. Better to generate a host immune response by having a vaccine. So let's see. Here's interferon mode of action. Is we've got the virus being taken up by a cell the cell then begins to produce interferon molecules, which is going to stimulate a set of antiviral proteins in another cell. And that will begin to block the viral life cycle from the second cell. So this is another defense system that we've got. So you end up, generating a variety of different kinds of systems that can inhibit the viral replication. The messenger RNA can be degraded. Protein synthesis can be inhibited. So interferons are able to arrange a number of different kinds of viral defense systems. Don't need to know about that too much, but if you're interested in it, it's more things to think about. This is the last slide, I think, for today. Yeah. So viruses can cause a whole range of tissue damage. They can destroy cells, like the herpes simplex virus is just blowing cells apart. HIV is seen here budding from white blood cells. It's going to weaken the cells by HIV infection. You can have cells being transformed, which we'll get into in just next time we talk. That is, having bits of DNA placed into their genomes that can start to cause them to become cancerous. These various sorts of inclusion bodies, Negri bodies that are in neurons infected by the rabies virus. The measles virus begins to produce these big gaps or syncytia in cells. All right, so our last discussion bit that we'll get into for viruses is all about cancer, and it's actually a really cool thing to get into. We'll pick up on this on Wednesday. All right, that's all I know. Thanks for your attention. Rock on. I'll talk with you later.